All right, so get ready because today we are going deep, deep into animal biotechnology.
Your Semester VI exam is coming up.
And we are here to help you absolutely ace it.
That's right.
Think of it like this whole deep dive
Uh-huh.
is like one giant super focused study session, walking you through the exact material you need to know to succeed.
Yeah.
And we have got an amazing treasure trove of questions and answers, taken straight from your BSc Biotechnology question bank 2 to be exact.
Yeah, we're not just skimming the surface here. We're going deep, deep, deep.
Absolutely. We want you to really, really understand these concepts
Understand them.
and be able to answer those exam questions with confidence.
Exactly.
So first up, let's imagine, just picture yourself in the lab.
Okay.
You're tasked with growing cells.
Ah.
What are the absolute must-haves, the things you absolutely cannot mess up?
You're talking about the physical and nutritional needs of cells and culture. So basically, we're trying to create that perfect artificial environment where the cells not only survive,
Right.
but they thrive.
Yeah, exactly. It's like creating a five-star hotel,
Yes.
but for cells.
I love that analogy.
So let's start with the ambiance of our cell hotel.
Okay.
Temperature.
Temperature. That's absolutely crucial. Mammalian cells.
Yeah.
The kind that we work with a lot in animal biotechnology, they're happiest at a cozy 37 degrees Celsius.
Yeah, makes sense. That's normal body temperature.
Exactly.
But why is that specific temperature so important? What happens if it fluctuates?
Well, imagine you're trying to bake a cake
Okay.
and the oven temperature keeps going up and down.
Mm,
What's going to happen to your cake?
disaster.
It's not going to turn out so well, right?
Right.
Same with cells.
Yeah.
Fluctuations in temperature
Yeah.
can really mess with their metabolism, slow down their growth,
Mm,
maybe even lead to cell death.
yeah.
And we have to think about
Right.
enzyme activity, protein structure,
Mm.
even how well those cells can stick
Oh.
to the surfaces they're growing on. All of this depends on maintaining that nice 37 degrees.
So consistent temperature is key. What about pH?
pH, just as crucial.
Yeah.
Think about it, you wouldn't want to swim in a pool
Right.
with a wildly fluctuating pH, right?
No.
Too acidic or too basic?
It's all about that balance.
Balance.
Yeah.
Mammalian cells, they like it slightly alkaline,
Okay.
between 7.2 and 7.4.
Okay. So how do we maintain this perfect pH? Isn't that where buffering systems come in?
Absolutely.
Yeah.
Buffers are key.
Yeah.
They help prevent those drastic shifts in pH that we don't want.
Okay.
And one of the most common systems used in cell culture
Mm-hmm.
is the bicarbonate system.
Okay.
But here's where it gets interesting.
Okay.
The bicarbonate system relies on CO2,
Wait, CO2.
carbon
I
dioxide.
thought that was like a waste product of respiration.
It is.
Yeah.
But it also plays a really crucial role in maintaining that pH balance.
Okay, so explain this to me. How does that work?
Okay, so when CO2 dissolves in the culture media,
Uh-huh.
it forms carbonic acid.
Okay.
And that carbonic acid is what helps buffer against those pH changes.
Oh, so it's all connected.
All about balance.
Balance, got it.
Yeah.
So temperature, pH, CO2, all under control.
Yes.
What about oxygen?
Oxygen, you're right.
We need that for respiration.
So most cells that we culture need oxygen.
Okay.
But here's where it gets really interesting, okay?
Okay.
Some cells actually thrive in low oxygen environments.
Really?
Yes.
Which ones?
Think about like deep inside tissues.
Okay.
Or even some types of tumor cells.
Wow.
So for those cells, we actually use special hypoxia workstations
Uh-huh.
that allow us to control the oxygen levels very precisely.
So we're mimicking their natural environment.
Exactly.
That's fascinating. What about humidity?
Humidity.
Why is that important in our cell hotel?
So humidity is all about preventing evaporation.
Okay.
In those incubators where we keep our cells, we aim for high humidity, usually around 95 to 98%.
Wow. Okay. Why is that?
Well, think about it. If the media starts to evaporate,
Uh-huh.
the concentration of all those solutes inside
Yep.
is going to increase.
Yeah.
And that can stress out our cells.
Oh, I see. So it's kind of like a plant wilting in the sun
Exactly.
if it doesn't get enough water.
High humidity,
Yeah.
happy cells.
Got it.
Now,
Yes.
this is the most important rule
It hit me.
of cell culture, all right?
Yeah.
Maintaining sterility.
Right. We don't want any unwelcome guests.
Contamination.
Is the enemy.
No bacteria, no fungi, no nothing crashing our cell party.
Exactly.
Yeah.
We want a pristine environment
Yes.
for those cells to flourish.
Okay, so that's where aseptic technique comes in, right?
Aseptic technique.
Yeah.
That's our weapon.
Okay.
It's a set of practices
Mm-hmm.
and procedures designed to prevent contamination
Okay, yeah.
at every step.
Okay, walk me through the basics.
What does aseptic technique actually involve?
Picture a surgical operating room.
Okay.
That's the level of cleanliness we're going for.
Yeah. Right.
We work in laminar flow hoods,
Yeah.
which have filtered air blowing across the work area constantly.
Keeping things super clean.
Super
Yeah.
clean. We sterilize all of our equipment,
Right. Okay.
all of our materials,
Yeah.
and of course we wear gloves, lab coats, and sometimes masks,
Yeah?
all about minimizing the chance of those microbes
Yeah.
getting into our cultures.
It's like creating a barrier between the outside world
Yes,
and our cells.
precisely.
Okay, and that brings us to antibiotics.
Antibiotics.
Yeah, they're like our little soldiers, right?
They're a common weapon
Fighting off those invaders.
in the fight against contamination.
So, I mean, it seems like a no-brainer, right?
Oh.
Just add them to the media.
It's not that simple.
Oh, okay, why?
Well, while antibiotics can be really helpful,
Mm-hmm.
especially when we're working with things like primary cultures. Okay.
Or when we're dealing with materials that may not be sterile,
Right.
we have to use them carefully
Okay.
because overuse can lead to antibiotic resistance.
Of course.
And that can create much bigger problems.
Yeah.
And plus,
Yeah.
some antibiotics can actually be toxic
Oh, right.
to certain types of cells.
Okay.
So it's a balancing act.
Got it. So it's not just about keeping things clean.
No.
It's about being smart
Yes,
about how we use those antibiotics.
exactly.
Okay, so what about monitoring those cultures? How do we know, despite all our best efforts, if contamination has snuck in?
We're like detectives, constantly on the lookout
Okay.
for clues.
Okay.
We check our cultures regularly
Uh-huh.
for any visual signs of contamination.
Okay.
So if we see cloudiness in the media,
Uh-huh.
unexpected color changes, or maybe even tiny little specks,
Okay, yeah.
that shouldn't be there.
Yeah.
Those are red flags.
Okay.
And if we suspect contamination,
Yeah.
we got to act fast.
Okay.
Typically, we'll discard that contaminated culture and start fresh.
All right, so better safe than sorry.
Right, especially because contamination
Yeah.
can spread
Yeah.
quickly.
And ruin experiments.
Ruin experiments.
Okay, so we've created this perfect sterile environment for our cells. The
Yes.
temperature, pH, CO2, oxygen,
Right.
humidity, and of course, that all-important sterility.
Sterility.
But what about the food?
The food.
Cells need to eat too, right?
Yes, they do.
Yeah.
They need nutrients to grow.
Okay, so what's on the menu?
That's where growth media comes in.
Okay.
It's like a carefully crafted recipe
All right.
that provides everything.
Yeah.
Our cells need to survive
Okay.
and flourish.
So what are the main categories on this cellular menu?
Well, broadly speaking,
Yeah.
we have two main groups,
Okay. Yeah.
natural media
Okay.
and artificial media.
Okay, so let's start with natural media.
Okay.
What makes them so natural? Well, natural media are derived from biological sources.
Okay.
Think blood serum,
Okay.
lymph,
Yeah.
or even extracts from tissues
Okay.
and embryos.
So it was like a little bit of everything.
Yeah, they were the go-to option
Okay.
back in the early days of cell culture
Okay.
when it was all just getting started.
Okay, so why were they so popular back then?
Well, because they offered
Yeah.
this really complex mix of nutrients,
Okay.
growth factors,
Yeah.
hormones,
Mhm.
all of that,
Yeah.
mimicking the natural environment that those cells are used to.
Okay, so they felt right at home.
Exactly. It
Yeah.
was like a rich,
Yeah.
nourishing soup
Okay, I can picture that.
for our cells
Yeah.
to thrive in.
Okay, but I imagine there were some drawbacks.
You're right.
Yeah.
Consistency was a major issue.
Okay, why?
Well, because the exact composition
Yeah.
of natural media could vary wildly
Okay.
from batch to batch,
Yeah, that makes sense because if you're taking it from different sources.
depending on the source.
Yeah.
It's like trying to follow a recipe
Yeah.
where the ingredients change every time you go to the store.
Right. Not ideal for scientific rigor.
Not at all.
What other drawbacks were there?
Well, ethical concern.
Okay, yeah.
Using animal-derived products
Right.
always raises those questions about animal welfare.
Of course.
Right.
Yeah. Yeah.
Plus,
Yeah.
there's always a risk of contamination
Right.
with viruses or other pathogens.
Yeah, coming from those animal sources.
Right.
So I imagine the quest for a more defined and ethical approach led to artificial media.
Exactly.
Yeah.
Artificial media was a significant leap forward.
Okay.
These are completely synthetic
Right.
formulations
Yeah.
where we carefully select
Okay.
and add specific components
Okay. Yeah.
to meet the needs
Right.
of our cells.
It's like we're designing a custom-made diet for our cells based on their specific
I
needs.
like that.
So it's less like a random soup kitchen
Yes.
and more like a 5-star restaurant
Yes.
with a carefully curated menu.
With a curated menu, exactly.
Yeah.
And this shift to a chemically defined composition brought some huge advantages.
Like what?
First of all, it eliminated that batch-to-batch variability
Okay, yeah.
that plagued natural media.
Right, because we know exactly what's going in
We know exactly
every single time.
what's in there.
Yeah.
So consistency, reproducibility.
That's a huge win for scientific research.
Absolutely.
So what other benefits did artificial media bring to the table?
Well, by moving away from those animal-derived products,
Yeah.
we address a lot of those ethical concerns
Absolutely.
that were associated with natural media.
Right.
Plus, it gave us much greater control
Yeah?
over the culture environment.
Okay.
We could fine-tune
Yeah.
the composition of that media,
Okay, so we could really optimize...
optimize cell growth,
Yeah.
differentiation,
Okay.
or even specific cellular functions.
Wow, that's really powerful. So artificial media sounds like the clear winner here.
Uh-huh.
But I'm guessing there's not just one type of artificial media.
You are absolutely right.
Yeah.
Artificial media come in many forms,
Okay.
each with its own advantages
Yeah.
and applications.
So what are those?
So we can think about serum containing,
Okay.
serum reduced,
Okay.
serum free,
Okay.
and chemically defined media.
Okay, let's unpack those, starting with serum containing.
Right.
I'm a little confused. Why are we still using serum
Yeah.
if we're trying to move toward that more defined approach?
That is a good question.
Yeah.
You see, serum,
Yeah.
despite its variability, is still a very potent source of growth factors
Okay.
and other components that a lot of cell types really need to thrive.
Okay.
It's kind of like a shortcut
Yeah.
to providing this complex mix
Right.
of nutrients.
Okay.
So it's kind of a trade-off between convenience and control.
Okay, so what would be an example?
So a common example
Yeah.
would be taking a basal salt solution,
Okay.
which provides like the basic salts and nutrients,
Yeah.
and then supplementing it with something like fetal bovine serum
FBS.
or FBS,
Okay.
typically at a concentration of 5 to 20%.
Okay, got it. Now, what about serum-reduced media? How are those different?
Serum-reduced media are kind of like a stepping stone
Okay.
towards serum-free culture.
Okay.
So the goal is to lower that serum concentration,
Okay.
usually down to between 0.5 and 2%,
Okay.
while at the same time adding in more defined components,
Okay.
like specific growth factors and hormones.
So we're kind of weaning them off.
We're trying to wean the cells off.
Yeah, that serum dependence.
They're serum dependent.
But still giving them the support they need.
Exactly, yeah.
So we're gradually increasing that control
Yes,
and reducing the variability.
absolutely.
Right, so now, the holy grail, serum-free media.
Ah, yes, the holy grail.
No serum at all.
Huh.
How on earth do those cells survive?
Well, it really is a testament to how far we've come
Yeah.
in understanding the needs of cells. We completely eliminate the serum,
Wow.
and we replace it with this carefully crafted cocktail
Okay.
of very specific chemically defined components.
So that means like growth factors.
Growth factors, hormones,
Yeah.
attachment factors,
Yeah.
lipids, you
Okay.
name it.
Wow.
It's all about providing exactly what they need
Okay.
and nothing more,
Nothing more, nothing less.
nothing less.
Okay. I mean, that sounds really complex to formulate.
Oh, it absolutely is.
Yeah.
Developing a serum-free media
Yeah.
is a meticulous process.
Right.
It requires lots of research,
Okay.
lots of optimization
Yeah.
to make sure that it meets those unique requirements
Yeah.
of each cell type.
Yeah.
But the benefits are huge.
What are they?
Increased control, reproducibility,
Okay.
and the ability to study
Yeah.
very specific cellular processes
Mm-hmm.
without interference from undefined serum components.
Okay. And finally, we have what sounds like the most basic chemically defined media.
Ah, CDM.
Yeah.
This is all about minimalism.
Okay.
It provides only the absolute essential components
Yeah.
needed for cell survival
Yeah.
and whatever specific function we're interested in studying.
Okay. So no frills, just the essential.
Exactly, just the basics.
When would you choose to use this approach?
This is typically reserved for highly specialized applications.
Like what?
Well, think biopharmaceutical production,
Okay.
where any undefined component could raise some red flags
Okay.
with regulators,
Right.
or maybe metabolic studies,
Okay.
where we need to track
Yeah.
nutrient uptake and utilization very, very precisely.
Right. So it's all about that ultimate control and precision.
Exactly.
Now, you mentioned some specific examples in the material. Can you tell us about a few of those key ones?
Absolutely.
Yeah.
Some commonly used artificial media
Mm-hmm.
include MEM,
Okay.
minimum essential medium,
Yeah.
DMM,
Okay.
Dolbeco's modified Eagle medium,
Uh,
RPMI 1640,
okay.
Ham's F12,
Okay.
and IMDM,
Okay, that's a mouthful.
Iscov's modified Dolbeco's medium.
Okay, but don't let those acronyms intimidate you,
No.
because each of these has its own characteristics and applications.
Yeah,
So
exactly.
it's like choosing the right tool for the job.
It is.
Yeah.
Depending on what cells you're working with
Yeah.
and what you are trying to achieve.
Can you give me some quick examples of how they're used?
Of course.
Okay.
So MEM, for example,
Okay.
was one of the earliest defined media.
Okay.
And it's still used today,
Yeah.
widely.
Yeah.
Especially for growing cells
Okay.
that like to attach
Okay.
to surfaces.
Got it.
Now, DM
Okay.
is a modification of MEM. And it's often the choice
Right.
for cells that divide rapidly
Okay.
because it has higher concentrations of amino acids
Mm-hmm.
and glucose.
Okay.
RPMI 1640.
Okay.
That one was originally developed
Oh, wow.
for culturing lymphocytes.
Uh-huh.
Those are the white blood cells
Yeah.
that are so essential
Yeah.
for our immune system.
Okay.
And it has a very unique buffering system.
Okay, so it's got some special features.
Yeah. Right.
What about Ham's F12?
Ham's F12
Yeah.
is known for its versatility.
Okay.
You can adapt it
Okay.
for a wide range of cell types,
Okay.
including some that are really hard to grow.
Really?
Yes.
Wow, that's cool. Okay, and then IMDM.
IMDM,
Yeah.
another modification of DM.
Okay.
And this one is often used
Okay.
for hematopoietic cells,
Okay.
which are the stem cells
Okay.
that give rise to all our blood cells.
Wow. Okay. So there's a whole world of media options.
There is.
Yeah.
There is.
And we need to be familiar with their strengths and weaknesses to make the best choice for our experiment.
Exactly.
Okay, so now let's move on to the next step in our cell culture journey.
Okay.
Let's say we want to study a very specific type of cell
Okay.
from a tissue sample.
Right.
How do we actually isolate and separate
Ah.
those precious cells from everything else?
That's where cell isolation and separation techniques come in.
Okay.
We have to carefully extract the cells we're interested in,
Mm-hmm.
and at the same time, preserve their viability and function.
Okay.
It's delicate surgery.
Okay, so paint me a picture.
Mm-hmm.
How do we actually go from this chunk of tissue
Yeah.
to a happy population of cells in a culture dish?
It's a multi-step process.
Okay.
First, we have to obtain that tissue.
Yeah.
Maybe it's a surgical explant,
Okay.
a biopsy,
Okay.
or even tissue.
Yeah.
From a cell bank.
Right.
Once we have our sample,
Yeah.
we have to break it down
Okay.
and release those individual cells.
That sounds messy. How do we do that without damaging them?
Well, we have two main approaches:
Okay.
enzymatic digestion
Okay.
and mechanical dissociation.
Okay, so let's start with enzymatic digestion.
All right.
What is that?
It's like using tiny little molecular scissors
Okay.
to cut through the glue that holds those cells together in the tissue.
So we're talking about enzymes like collagenase and trypsin, right?
You got it.
Yeah.
They break down that extracellular matrix.
Yeah, that scaffolding that surrounds the cells.
Exactly.
Yeah.
Now, by using specific enzymes,
Yeah.
or even combinations of enzymes, we can really tailor that digestion process
Okay.
to different tissue types.
So, for example, if we're trying to break down collagen.
Collagenase is
Yeah.
a good choice.
Okay.
It's a major component of connective tissues,
Right.
while trypsin
Okay.
is more effective at breaking down the proteins that actually hold those cells together directly.
Okay, so that's enzymatic digestion.
Hmm.
What about mechanical dissociation?
Mechanical dissociation relies on physical force
Okay.
to separate those cells.
Okay, what does that involve?
Think scraping,
Okay.
mincing,
Okay.
sieving,
Okay.
or even homogenization, where we blend the tissue
Yeah, okay.
into a uniform suspension.
That sounds a little rough.
Yeah, sometimes it is.
Yeah, when would you choose this method?
Well, mechanical dissociation
Yeah.
is often preferred
Yeah.
for tissues that are less sensitive
Okay.
to enzymatic damage,
Right.
or when we need a really rapid cell isolation.
Okay.
It's also useful
Yeah.
for tissues
Yeah.
that are tough to digest
Okay, yeah.
enzymatically, like
Yeah.
muscle or bone.
Right, okay.
Yeah.
So we choose our weapon based on the type of tissue and how sensitive those cells are?
Exactly.
But once we've broken down the tissue,
Yeah.
we don't always want every single cell type.
You're right.
Yeah.
Most tissue. Yeah.
Contain a mix of cell types.
Right.
And we often want to isolate very specific populations.
Okay. Yeah.
So that's where cell separation and enrichment techniques come in.
Okay. Yeah.
It's like panning for gold.
Okay.
We need to sift through all that dirt
Right.
to find those precious nuggets.
Yeah.
The cells
The cells that we want.
we're interested in.
Okay, give me the highlights. How do we actually separate these cells?
We have a whole arsenal of techniques.
Okay.
One common one is differential adhesion,
Okay.
which exploits the differences in how strongly different cell types will stick to surfaces.
Okay.
So by carefully choosing the right surface,
Okay.
we can actually encourage some cells to attach
Okay.
while others remain in suspension.
So it makes it easier to separate them.
Make separation easier.
Yeah, clever. So we're kind of playing with their preferences.
Yeah,
Okay.
we are.
What else?
Another powerful technique is density gradient centrifugation.
Now, for this one, we create a gradient
Okay.
of a dense solution,
Cool.
like Fickle or Percol.
Okay.
We layer our cell suspension on top,
Okay.
and then we spin it really, really fast.
In a centrifuge.
In a centrifuge.
Right.
And the cells will migrate through that gradient
Okay.
based on their density.
Oh, okay. So it's like creating layers of a parfait
I like that.
Yeah.
With different cell types settling
at different levels.
based on their density.
Exactly. That's really cool.
Now, this technique is great
Yeah.
for separating cells based on their size and density.
Okay, cool.
And then
Yeah.
for even more precise cell sorting,
Okay.
we have the big guns,
Hit me.
FACS,
Okay.
fluorescence-activated cell sorting,
Okay, that one.
and MACS.
Those are the ones I was waiting for.
Yes,
Yeah.
magnetic activated cell sorting.
Can you remind me how those work?
Okay, so imagine this. With FACS, we use fluorescently labeled antibodies to tag specific cell surface markers.
Okay.
So we label those cells,
Mm-hmm.
pass them through a laser beam, single file,
Okay.
and a detector analyzes the light that is scattered and emitted
Oh.
by each cell.
Okay.
Based on that fluorescent fingerprint,
Yeah.
we can sort individual cells
Wow.
into different populations.
So it's a microscopic laser show
It is.
with cells zipping through.
Zipping through
Yeah.
at lightning speed.
Yeah. That is mind-blowing. What about MACS?
MACS, it's kind of like fishing for cells
Okay.
with tiny magnets.
Okay.
We use magnetic beads coated with antibody
Mm-hmm.
that bind specifically to our cells of interest.
Okay.
So then
Yeah.
we pass this whole mixture through a magnetic field.
Okay.
The labeled cells stuck to those beads get trapped,
Okay.
while the unlabeled cells just flow right through.
Separation achieved.
Voila.
That is so cool. I mean, it's incredible how we can manipulate cells at such a tiny scale.
It is.
I mean, those both sound incredibly powerful for research and clinical applications.
They are essential
Yeah.
for isolating and enriching those specific cell populations.
Yeah.
For everything from basic research
Right.
to cell therapy
Oh.
to regenerative medicine.
Wow.
Yeah.
Okay, so we've successfully isolated our target cells.
We have
What's the next step?
the next step.
Don't we need to know how many cells we have?
You're reading my mind.
Yeah.
That's where viable cell counting comes in.
Okay.
As fundamental to cell culture as flour is to baking.
I like that analogy.
We need to know how many living cells
Yeah.
we have in a given sample.
So it's like taking attendance.
It is like taking attendance
For ourselves.
for ourselves.
Yeah, I like that.
And this number
Yeah.
is crucial for a whole range of applications.
Like what?
So first,
Yeah.
it lets us determine the cell density
Oh.
and viability.
Okay.
So how many cells we have
Yeah.
per unit volume. And what percentage of those cells are actually alive.
Right, because we want to know if we're starting with a culture that's half dead.
Yeah.
Right?
Yeah,
We need to know that they're healthy and ready to go.
absolutely.
Okay, so it's a health check
It is.
for our cells.
It is. And it's essential for monitoring cell proliferation over time.
Okay.
So by tracking those changes in cell number,
Yeah.
we can plot growth curves,
Okay.
essentially visualizing how quickly our cells are multiplying
Right.
and how they're responding to maybe different treatments.
Right, or experimental conditions.
Or different conditions, exactly.
So we can see how they're growing, kind of like a population.
Yeah.
over time.
It's like watching a
Yeah.
population grow.
Yeah. And you can see how different factors are affecting that growth.
Exactly.
Yeah.
And remember when we talked about standardizing cell seeding?
Yeah, making sure they're all starting at the same starting line.
Yes.
Yeah.
Well, accurate cell count
Yeah.
is essential for that.
Right.
It ensures that any differences that we see down the line
Right.
are due to our manipulations.
Not the starting cell density.
And not just variations.
Yeah, exactly. Okay.
Well, it's also incredibly valuable for evaluating
Yeah.
the effects of things like drugs.
Yeah.
Or other treatments,
Yeah.
particularly cytotoxic effects.
Oh, okay. Yeah.
So by comparing the cell counts of treated and untreated samples,
Yeah.
we can actually quantify how those treatments
Right.
are affecting
Yeah.
cell viability.
So basically how toxic something is
Exactly.
to our cells.
Yes.
Okay.
And finally,
Yeah.
viable cell counting is used in a whole range of cell-based assays
Okay.
to normalize data.
Okay.
So by accounting for any differences in cell number,
Okay.
we can make much more accurate comparisons between different groups.
So it's like adjusting for population size when you're comparing data
Yes,
between different cities.
And-
You have to take that into account.
absolutely.
Yeah.
Viable cell counting is truly a cornerstone
Yeah.
of cell culture work.
Okay.
And mastering these techniques
Yeah.
is essential for anyone
Yeah.
working in the field.
Okay, so now that we've revisited the why of cell counting,
Yes.
let's talk about the how.
Okay.
Can you remind us the main methods for performing these counts?
Absolutely. So, as we talked about before, we
Yeah.
can broadly divide them into two categories.
Okay.
Dye exclusion methods
Right.
and metabolic activity assays.
Okay.
Yeah.
Okay, so let's do a quick refresh on dye exclusion methods.
Okay.
How do they work again?
So, dye exclusion methods take advantage of the fact that certain dyes
Okay.
can only enter cells
Right.
that have damaged cell membranes.
Okay.
So they selectively stain the dead cells,
Yeah.
leaving the live cells unstained.
It's a simple way to tell the difference between the living and the dead.
Yes, it is.
Yeah.
It is.
And the most common dye is
Trypan blue.
trypan blue.
That's the go-to dye
Yeah.
for a lot of researchers.
Right.
It's inexpensive,
Right.
it's readily available,
Mhm.
and it's easy to use.
So how do we actually use it?
We mix our cell suspension
Okay.
with the trypan blue,
Mm-hmm.
load it onto a hemocytometer,
Yeah.
which is a specialized counting chamber,
Right.
and then we count the stained and unstained cells
Under the microscope.
under the microscope.
So it's a manual counting method.
It is.
What are the downsides there?
Well, as you can imagine,
Yeah.
manual counting can be pretty time-consuming.
Yeah. Especially if you have a lot of samples.
Especially if you have lots of samples.
Right.
And it can be subjective, right?
Right.
Different people
Yeah.
might count the same sample
Yeah.
a little differently.
Yeah.
So that introduces variability.
Okay. Are there any alternatives to this manual counting with trypan blue?
We could use another dye
Right.
called erythrocin B.
Erythrocin B. Okay.
Works similarly to trypan blue,
Okay.
stains the dead cells,
Mm-hmm.
but it's a different color,
Oh, okay.
which some researchers prefer.
A different shade of death indicator.
You could say that,
Yeah. Okay.
but if we really want to eliminate that subjectivity
Yeah.
and speed things up,
Yeah.
we can turn to automated cell counts.
Yeah. Those fancy machines that are in all the labs.
Those fancy machines.
How do they work?
There are two main types,
Okay.
Coulter counters
Okay.
and image-based automated cell counters.
Okay.
Coulter counters
Yeah.
work by measuring changes in electrical resistance.
Electrical impedance. That sounds complicated.
It sounds complicated.
Break it down for me.
Basically, imagine
Okay.
a tiny little electrical gatekeeper
Okay.
for our cells.
Right.
As cells pass through a small aperture
Uh.
in the Coulter counter,
Okay.
they change that electrical resistance.
Yeah.
So the machine measures those changes
Oh, okay.
to count the cells.
So each cell creates a little blip.
A little blip.
Yeah. Okay.
And it can even give us information about their size and volume,
Oh, wow.
because different cell types have different sizes and electrical properties.
Okay.
Coulter counters can sometimes even distinguish
Wow.
between those different cell populations.
That's amazing.
Hmm.
What about the image-based automated cell counters?
Image-based counters use digital microscopy
Okay.
and some pretty clever image analysis algorithms
Yeah.
to identify
Yeah.
and count those cells.
Okay.
They're taking pictures of the cells
Uh-huh.
and then using software to analyze those images,
Okay.
counting the cells.
Yeah.
But they can even tell us information about their size, their shape, and other features.
So it's like having a microscopic eye
It is.
that can see and analyze our cells
Yes.
with amazing precision.
Yes.
That's incredible. So these automated counters have really revolutionized cell counting.
They have.
Yeah.
They're fast, they're accurate,
Yeah.
and they're objective.
Right. No more human error.
No more human error.
But I'm guessing they're pretty pricey.
They can be a significant investment.
Yeah.
But for labs that do a lot of cell counting,
Yeah.
it's worth it.
Right. It's an investment. Okay. So we've got our dye exclusion methods. We've got our automated counting.
Yes.
Let's talk about those metabolic activity assays again.
Okay.
Can you remind me how those work?
So metabolic activity assays offer an indirect way
Okay.
to assess cell viability.
Okay. So instead of directly counting.
Instead of counting, we're
Yeah.
measuring
Okay.
their metabolic activity.
Right. So if the cells are active,
Yes,
they're alive.
exactly.
Okay.
One common type of metabolic activity assay uses tetrazolium salts.
Okay.
Those are compounds
Right.
like MTT,
Okay.
MTS,
Uh-huh.
XTT,
Okay.
or WST1.
All right. So those are our options.
Those are some options.
Yeah.
They're colorless
Okay.
in their oxidized form,
Okay.
but
Yeah.
metabolically active cells have reducing power,
Okay.
so they can donate electrons.
Okay.
So when we add these salts to our cells, they get reduced,
Okay.
and they're converted into colored products.
Oh, I see. So it's a color-changing test
Osteosis.
for activity.
Yes.
The more active,
The more active,
the darker the color.
the more intense the color.
Okay.
Yes.
And we can measure that.
And we can measure that very easily
Okay.
with a spectrophotometer.
Okay.
Now, another widely used metabolic activity assay
Okay.
is the Resargerin assay.
Okay.
Resargerin is a blue non-fluorescent dye
Okay.
that gets reduced
Okay.
by those metabolically active cells
Okay.
to resorphin,
Okay.
which is pink
Okay.
and fluorescent.
Oh, cool.
So we can measure
Yeah.
either the color change
Right. Blue to pink.
or the increase in fluorescence.
To get a measure of how active they are.
Exactly.
That's clever. Okay. So we have all these different methods
We do.
for counting cells.
Yes.
How do we pick the best one?
Choosing the right method
Yeah.
really depends on a number of things.
Okay.
The type of experiment,
Right.
the level of accuracy that we need,
Okay.
the throughput, how many samples we have,
Right.
and of course the resources that we have available.
Makes sense. So match the tool to the job.
Exactly.
And regardless of which method we pick,
Yes,
careful sample preparation,
absolutely.
optimizing the conditions,
Yes,
and thinking carefully about those results
absolutely crucial.
to make sure that we're getting reliable data.
Reliable data.
Okay, that was a great overview
Thank you.
of viable cell counting. It's a pretty fundamental technique for anyone working with cell cultures.
It is.
Now let's talk about a technique that lets us manipulate the cell cycle and study those cell cycle dependent events in a very controlled way. Cell synchronization.
Cell synchronization.
Yeah.
It's a fascinating technique
Yeah.
that allows us to hit the pause button
Okay.
on the cell cycle
Okay.
and essentially synchronize cells
Okay.
to a specific stage of the cell cycle.
So we're getting them all to do the same step
Yeah,
in the dance
Yeah.
in unison.
exactly.
Okay, I like that.
By doing so,
Yeah.
we can gain incredible insights into what's happening during each of those stages
Okay.
and study processes that are really tightly coupled to those specific phases.
Okay, before we get into the how,
Yeah.
remind us quickly the what,
Yeah.
the different stages of the cell cycle.
The cell cycle, as you may recall,
Yes.
is that series of events that leads to cell growth and division.
Right.
And we typically divide it into 4 main phases.
Right.
G1.
Okay, first gap phase,
Yeah.
DNA
Or
replication.
Q2.
Second gap phase,
And BIM.
mitosis.
Fantos. Yeah.
So G1,
The cell is growing, getting ready for DNA replication.
growing,
Yeah.
prepping for that DNA replication.
ES phase is where all the magic happens.
DNA replication.
Yeah, the DNA is copied.
Yeah.
G2, it's growing some more.
Still growing.
Yeah.
Getting ready for division.
Getting ready for the big split.
Yes.
Yeah.
And then M phase.
Yeah.
Mitosis.
It divides.
The cell divides.
The grand finale,
Grand finale.
2 daughter cells.
Yes.
Okay, perfect. So cell synchronization lets us pause this intricate dance at very specific points.
Specific points.
Yeah. Why would we want to do that?
Well, there are a number of reasons.
Okay.
So first of all,
Yeah.
it's really valuable for studying cell cycle progression and regulation.
Okay.
So by synchronizing cells,
Uh-huh.
and then releasing them from that arrest,
Okay.
we can actually track them as they move through the cycle.
Okay.
Identify those checkpoints
Right.
that control the transitions.
Right. Between the phases
Yeah.
and study the factors that influence
Okay.
the pace
Right.
and timing of the cell cycle.
So it's like setting a starting pistol
Yes.
and then watching those
We're
runners
watching those runners.
go through the course.
Yes.
Yeah. And we can see how fast they are at different points
Exactly.
and how their stamina changes.
Exactly.
That's really cool. What else?
It's also crucial for analyzing
Okay.
cell cycle dependent gene expression.
Oh, okay.
So,
Yeah.
many genes are only expressed during specific phases of the cell cycle.
Right. So different genes for different steps in the dance.
You got it.
Okay.
So if we synchronize those cells
Yeah.
to a particular phase,
Okay.
we can isolate the RNA or protein from those cells
Yeah.
and analyze
Yeah.
the expression patterns of those genes.
So that gives us a molecular understanding of how that cell cycle is controlled.
Yes, it does.
That's fascinating.
Hmm.
What else?
It's also really helpful
Yeah.
for studying
Yeah.
DNA replication
Okay.
and repair mechanisms.
Okay. So DNA replication, we know that happens in the S phase.
Exactly.
Yeah.
So if we want to study how the DNA is copied,
We need to synchronize them to the S phase.
yes.
Okay.
And we can also synchronize them to different phases
Yeah.
to study how they repair
Right.
that damage.
Okay. So it's about having that temporal control
It is.
over these really important processes.
Yes,
Are there any other applications?
absolutely.
I mean,
It's also used to analyze mitosis
okay.
and chromosome segregation.
Okay.
That's-
Mitosis, that's the big one where the chromosomes are separated.
Yes, the chromosomes condense,
Yeah.
the spindle forms,
Yeah.
and the chromosomes are separated
Yeah. The grand finale
into those two daughter cells.
where everything has to be perfect
Exactly.
to make sure those daughter cells get all the right stuff.
Everything in the right place.
Yeah. Okay.
So by synchronizing cells to M phase,
Yeah.
we can really study that machinery
Yeah.
in detail,
Okay.
identify potential errors
Right.
that might lead to problems like cancer,
Okay.
and even screen for drugs that might target
Okay. Yeah.
those processes.
Because if you can stop them from dividing, you
Exactly.
can stop cancer.
Exactly.
No.
That's especially relevant
Yeah.
for anti-cancer therapies.
Okay. So it's about really understanding that phase
It is.
and how it works and how we can target it.
Yes.
Okay. What else?
Cell synchronization is also used
Yeah.
in drug screening
Okay.
and sensitivity assays.
Right.
So, as I mentioned,
Yeah.
a lot of anti-cancer drugs
Yeah.
specifically target cells
Right.
in certain phases of the cell cycle.
So we synchronize them to that phase and then we test the drug,
Exactly.
see how effective it is.
To see if they're working,
Yeah.
if they're effective at killing those cancer cells, but not harming the normal cells.
That's amazing.
It's a powerful tool
Yeah.
for preclinical drug development.
Okay. So that was drug screening. Anything else?
And finally,
Hit me.
we can use cell synchronization to study
Okay.
cellular differentiation
Okay.
and development.
Okay. Explain that one to me. How does that work?
Cellular differentiation
Yeah.
is that process where cells become specialized
Okay. Yeah.
to perform very particular functions.
Right. Like stem cells turning into all the different types of cells.
Yes. Think about how a stem cell can give rise to all those different cell types in our body.
Yeah. That's amazing.
Muscle cells,
Yeah.
nerve cells,
Yeah.
blood cells,
Yeah.
you name it.
Yeah.
So this differentiation process often involves changes
Okay.
in gene expression
Right.
and cell cycle regulation.
Okay.
So by synchronizing cells
Mhm.
and then inducing that differentiation,
Okay.
we can track those changes
Yeah.
over time and gain a better understanding of what's happening.
So we're basically watching them transform
Yeah.
from those generalists
Yes.
to the specialists
To the specialists. And they're following that carefully choreographed program.
of gene expression and cell cycle control.
Exactly.
That's incredible. So as you can see,
Yes.
cell synchronization is this really powerful tool
Powerful tool.
with a huge range of applications.
Yes.
It can be used for everything from basic cell biology
Yes.
to drug discovery
Yes.
to developmental biology.
Absolutely.
This is amazing. We can manipulate these cellular dances with such precision
We can.
using all these different techniques.
Yes.
Okay, but with all these methods, are there any limitations
That's a great question.
or things we should be careful about when we're using cell synchronization?
Yeah, cell synchronization
Yeah.
is a powerful tool,
Hmm.
but it's not without caveats.
Okay.
So No synchronization method is perfect.
Okay, so explain that to me.
There will always be some degree of variability
Okay.
in how individual cells
Yeah.
respond
Right.
to these treatments.
So you're not going to get perfect synchrony
No, not perfect.
across a whole population of cells.
Right.
Right. So it's kind of like trying to get a large group of people to walk in perfect step.
Yes,
You're going to have some stragglers.
you will.
Yeah.
Okay. What else?
We also need to be aware of potential artifacts,
Okay.
those unintended consequences
Okay.
that can come up from our manipulations.
Oh, yeah. Give me an example.
Well, for example,
Yeah.
prolonged cell cycle arrest
Okay.
can sometimes actually alter cell physiology
Oh, wow.
or G expression patterns.
Right.
So,
Yeah.
it's really important to choose a method
Mm-hmm.
that's appropriate
Yeah.
for the specific cells and the research question.
Okay, so minimize the stress.
You minimize the stress
Yeah.
and keep it short.
Keep it as short as possible.
Yeah, okay, so don't stress them out too much.
Exactly.
All right, what other things do we need to keep in mind?
Validation
Okay.
is key.
Okay.
We need to make sure
Yeah.
that the method we choose is actually working
Right.
the way we expect it to, and it's
Right.
not
Yeah.
causing damage
Right.
or introducing any artifacts.
Right. We don't want to measure something that's not real.
Exactly.
So how do we validate?
Well, there are a few things we can do.
Okay.
Flow cytometry,
Okay.
that laser-based cell sorting method we talked about,
Yeah.
can help us assess the efficiency
Okay.
of synchronization.
Okay. And we can also use microscopy, right,
We can also use microscopy
to look at the cells.
to look at the cell,
Make sure they look healthy.
make sure they're happy.
Yeah. Okay.
And finally,
Yeah.
it's important to keep in mind
Yeah.
that the effectiveness
Yeah.
of different methods can vary
Yeah, okay.
depending on the cell type. Oh, okay.
Some cells are more amenable.
So some cells are more stubborn than others.
Some are more stubborn.
Yeah.
Some might be more sensitive
Okay.
to certain drugs.
Right.
Their cell cycle kinetics might be different.
Okay.
So it really is
Yeah.
a bit of trial and error
Okay. So we really have to get to know ourselves.
to find the best method
Yeah.
for a particular cell type and research question.
Okay. So it's all about choosing the right tool for the job,
It is.
optimizing the conditions,
Absolutely.
validating those results.
Yeah.
To make sure we're getting good data.
Good, reliable data.
Okay, that was a great deep dive
Thank you.
into cell synchronization.
Hmm.
It's really amazing how we can manipulate those cellular dances with such precision.
It is remarkable.
So we've explored the physical and nutritional needs of cells in culture,
Mm-hmm.
the techniques for isolating and separating them,
Yes.
the art of counting them, and the
Everything.
power of synchronizing them.
Yes.
Yes.
What a journey.
It has been quite a journey.
Any final thoughts for our listeners as they get ready for this big exam?
Well, I think the key takeaway here
Yeah.
is that cell culture is really a fundamental tool
Mhm.
in animal biotechnology.
Okay.
And mastering these techniques,
Yeah.
understanding those principles, the applications, the limitations,
Mm-hmm.
that's what sets you up for success,
Absolutely.
both in your exams
Yeah.
and in your future careers.
Right. It's not just about memorizing the facts.
No.
It's about understanding the concepts,
Yes.
connecting the dots,
Connecting the dots
Yeah.
and seeing how everything fits together.
seeing the bigger picture.
A bigger picture.
Okay, now, before we wrap things up,
Yes,
is there one final thought-provoking question?
absolutely.
Something to really get them thinking.
We've covered a lot today,
Yeah, we have.
exploring those fundamental principles of cell culture.
Mm-hmm.
But I want to challenge you to think about this.
Okay.
As our understanding of cell biology deepens
Yeah.
and our technologies become more and more sophisticated,
Right.
what new possibilities do you envision for the future of animal biotechnology?
Oh, that is a great question. It's all about pushing those boundaries.
Pushing the boundaries.
Yeah. And imagining what's possible.
Imagining what's possible.
Maybe some of our listeners will be the ones making those breakthroughs.
I have no doubt.
Yeah.
The future of animal biotechnology is bright,
Yeah.
and it's in the hands of curious and innovative minds like yours.
Well said. Thank you so much for joining us on this deep dive.
It was my pleasure.
I hope everyone feels empowered and ready to conquer that Semester VI exam.
Good luck, everyone.
Best of luck to all of you on your exams
Yes.
and in your future endeavors
Absolutely.
in this amazing world of biotechnology.
It's a fascinating world.
Until next time, keep those cells happy.
Keep them happy.
Keep those minds curious.
Curious.
And as always, happy studying.
Happy stay.